For our CCNB Data Wednesday talk, I'm really excited to introduce Dr. Kelley Harris, who will be sharing her research with us today. Kelley got her PhD in applied mathematics and computational biology at the University of California, Berkeley, and she was an NIH NRSA postdoctoral fellow at the Stanford University Department of Genetics in the School of Medicine prior to her current position, an assistant professor in the Department of Genome Sciences at the University of Washington. Her computational biology research group uses large data sets of genetic variation to study the evolutionary history of humans and other species, with particular interests in coalescent theory and the evolution of the mutation rate and spectrum. And with that, I'll let Kelley take over.
Oh, thanks, Kelsey, for the invitation and for hosting me. It's nice I wish I could be there in person with you, but it's been nice to see so many familiar faces and meet new people on this visit. So I'll try and share my screen now. Oh, great. So today I'm going to talk to you about a couple of different stories in my lab that are have in different systems, but both revolve around discovery of natural mutator alleles, which is really exciting. It's sort of what we'd been hoping to do from the founding of my lab 3 years ago, and are finally starting to be able to do in practice. So just jumping right into the first story I have to tell you about, my lab has been fortunate enough to work with a really unique data set that's kind of like a modern spin on a very old and very powerful mouse data set. So basically, back in 1971, the lines called BXDs or B by Ds were originally founded by taking the standard laboratory mouse C57 Black 6 and crossing it to a different laboratory mouse strain called DBA2J. And after obtaining some F2s from this cross, a bunch of different lines were founded and inbred by brother-sister mating. And this proved to be so useful for quantitative trait locus mapping that 5 more cohorts of these B by D mice were founded in later years, up until the most recent cohort being founded just a few years ago in 2014. And so, yeah, as I alluded, the reason that these cohorts keep getting maintained and expanded is that they're very useful for mapping the architecture of complex traits. And it was sort of fun for me to read a bit of a survey of this types of traits that have been mapped using the B by Ds, things ranging from cadmium-induced testicular damage to various kinds of drug and alcohol dependence. It's possible to study these things in this system with maybe more precision than you could in humans by just taking a bunch of these lines that have this very well understood genetic relatedness structure, and then exposing them all to some stimulus like drug or alcohol or some environmental perturbation, and then seeing whether if you observe a difference in how the different B by Ds respond to the stimulus, whether there are any loci in the genome where having Black 6 or DBA2J ancestry predicts the phenotype. So about over 3 years ago now, as I was finishing up my postdoc at Stanford, my postdoc advisor, Jonathan Pritchard, and I had the idea of basically sequencing all of these strains to get at a phenotype we were especially interested in, which was the germline mutation rate. And I'll get into in a minute why these lines are so well suited for mapping that trait. But so together with Rob Williams, who is the founder of one of the later strains, and currently houses a bunch of these mice at the University of Tennessee Health Center, as well as Abe Palmer, who started working on these when he was up at the Jackson labs. Basically, we all got together to form a little consortium to sequence all the B by Ds to 30x coverage. Sort of an fun aside is that we decided to sequence them with 10x genomics technology in hopes of mapping some copy number variation, but the sort of SNP calling tool that works with the long range, with the 10x tags ended up being so slow that we sort of had to throw away this extra information for now and just call SNPs using GATK. So yeah, if anyone has sort of found better ways to actually deal with 10x SNP calling, we'd love to hear about them. But basically, what we were interested in doing was going through all of these recombinant offspring and recording the locations of homozygous singletons, so variants that are homozygous in that strain and absent from all the others, because these are most likely to have occurred as de novo mutations sometime between when the strain was founded and the present. And these are mostly homozygous just because of the maintenance of the strains by brother-sister inbreeding that causes, can cause a new mutation to fix in as little as one generation. And so my great postdoc, Tom Sassani, has been working on this for about the past year, and just started by calling all these homozygous singletons and counting up how many occur in each of the strains. And the result that we expected to see and luckily did see was that the oldest strains that were founded in the 1970s have a lot more homozygous singletons than the younger strains founded just a few years ago, which you can see here. So this is a distribution of just the number of homozygous singletons within and across each cohort. And I forgot to say, feel free to interrupt me with questions at any time if people would like to. So this gave us some confidence that we are seeing the relics of mutations as we expect to. And so we estimated mutation rates in each strain. Oh, this is just to say, this is kind of similar to what people do calling de novo mutations in parent-child trios. When you say sequence an individual and their parents to find the locations of the new mutations that have occurred in the child. But when you do that, you get less than 100 typically mutations per trio. So you just get a lot noisier estimates of mutation rates on each genetic background than you do in this case, where you sort of keep propagating the lines and just get a lot more mutations per fixed genetic background. Another thing is that since de novo mutations are heterozygous, and there's often some uncertainty about which ones sort of are true variants and which are maybe somatic variants segregating at like a lower than 50% frequency in the blood. Like there's a lot more uncertainty of counting de novo mutations subject to these bioinformatic errors than we think we have in this nice case where everything is homozygous. And so to calculate the mutation rate of each BXD strain, we just took the number of homozygous singletons divided by the generations of inbreeding, which has been recorded by the mouse caretakers, and then divided by the size of the accessible genome. So the size of the genome minus the regions masked due to repeat content and such quality issues. And so then you see there's some variation, but a lot of the variation between epochs goes away. And just for comparison, there have been previous estimates of mouse mutation rates from standard trios and other pedigrees. And what we get is sort of centered at the previously estimated range, which was reassuring. And what we really want to know is how much of this variation in mutation rate might be heritable by genes that, or factors that we could have some hope of mapping with this cohort. So one reason that we think there might be sort of genetically encoded differences in mutation rate between C57 Black 6 and DBA2J is this previous paper by Beth Dumont, which looked at the mutation spectra of these among other lab mouse strains. And so what I'm showing you here is a figure from her paper where each mouse is associated with a bar plot showing the composition among sort of young mutations in this lab strain of mutations of the 6 basic types, C to A, C to G, and so on, just collapsed with reverse complements regarded as the same thing. And so you can see from this that DBA2J appears to have a bit more C to A mutations and a bit fewer C to T mutations compared to C57 black 6. And whenever you see a difference in mutation spectrum like this, what we suspect it means, and it's a thing that's been confirmed mostly in cancers, is that there's some mutational process working with a higher rate in one strain than the other, which tends to create certain types of mutations more often than others. And so having a different spectrum is sort of indirect evidence that mutations are being generated in different ways in these 2 strains, and they very likely could have different mutation rates as well. And so this is data from recent mutations that occurred in lab environments. There's been some question in human trio data about whether differences between, say, populations like the Amish and others are really due to genetics or due to diet and the rest of the environment. And that's less likely in these lab mice that by and large get fed the same things and reared in the same ways. Also, what you can sort of see if you look at like the similarity in names, and the similarity in spectra of columns that are next to each other, or that mutation spectra of strains that are a bit more closely related to one another, are a bit more similar than random strains. And that's what you might expect if this is some kind of heritable phenotype. And so this is a PCA plot from Beth Dumont's paper on the left compared to a PCA plot of mutation spectrum variation within humans on the right. And basically this work in humans that Jonathan and I were doing around the time that we started planning this mouse project. Basically what we've seen is that when you partition mutations by triplet context, so then you have 96 types of mutations depending on what are the bases on the immediately to the left and right of the base being mutated. You see a slightly different mutation spectrum in each human continental group, suggesting like if some of this is genetic, that like there are modifiers of the mutation rate and spectrum segregating in humans. And what Beth saw in the mice was something reminiscent of this, of like a couple of principal components of significant mutation spectrum variation among the mouse strains. And including in green, the DBA family having different mutation spectra from the C57 family in pink. And basically the, like what, as I said before, what recombinant inbred lines like the BXD were designed for was to facilitate QTL mapping, where you measure a trait in a bunch of these organisms and then do sort of like a GWAS across association study across the whole genome to look for peaks where mice with one ancestry background are significantly different in the trait than the other. And so when we did this for the total mutation rate that we estimated from the homozygous singletons, we unfortunately didn't get any genome-wide significant hits. But then we looked at traits that were features of the mutation spectrum. And then we did find one that was associated with specifically the rate of C to A mutations. It's a big genome-wide significant peak on chromosome 4. And this peak is even more pronounced when instead of the C to A rate, what you use as your trait is the C to A fraction. So just the fraction of singletons in each strain that are C to A's rather than other mutation types. And this one QTL peak, I guess a little funny to call it a quantitative trait locus when like we only see one, but it is still a quantitative trait, each of these mutation summaries. Basically, what I'm showing on the left here is how the C to A mutation fraction among singletons varies between strains with a homozygous DBA2J haplotype at the QTL versus a heterozygous QTL versus a homozygous C57 black 6 haplotype. We'll just sometimes shorthand call these like the B and D haplotypes at the QTL. And then, so if you do a PCA of the mutation spectra of all the BXDs and then color them by whether they have the D or the B haplotype, it pretty neatly explains the first principal component of the variation, which you can see like on the bottom loadings panel is largely dominated by the C to A fraction. And we also examined the triplet context distribution of the C to A mutations that are occurring at a higher rate in the individuals with the D ancestry. And what we see is that it does have a specific context bias where mutations from CA to AA and mutations from CT to AT are that's the type that is the most enriched in the mice with the D haplotype. And this is just a heat map showing all 96 types of mutations that you get sort of by looking at the 5' and 3' bases adjacent to the SNP, and how the sort of two red stripes you see in the C to A box at the top are the odds ratios enrichments of these mutation types in the mice with the D versus the B haplotype. And so, okay, now that we have this nice QTL, it's still a fairly large interval of about 5 megabases on chromosome 4. So we really wanted to see what genes are in here, like how close can we come to really fine mapping what's causing this QTL. And so it turns out this region contains 100 different protein coding genes, but then Tom first filtered these by which contain variants that are annotated to have moderate or high impact by a program called SNP-F. And that narrowed this down to 22 genes. And then we filtered further to genes annotated by gene ontology as being involved in DNA repair and replication. And that actually narrowed it down to only one gene called MUTYH, which we were really excited about because MUTYH is involved in the base excision repair pathway that prevents C to A mutations that are caused because of oxidative stress, where guanine becomes oxidized to 8-oxoguanine. So it turns out that when this particular lesion occurs and is not repaired, that tends to result in C to A mutations. And so this is just a little cartoon about why MUTYH is necessary to repair this DNA damage that's caused by oxidative stress. And we found that there has been some work on what happens if you knock out MUTYH, both in mouse embryonic stem cells and in the germline of live mice. And so there's evidence from this prior work that knocking out MUTYH does elevate the germline C to A mutation rate. And moreover, there's this paper by Ono et al., which they generated these mice with the entire 8-oxoguanine repair pathway knocked out, MUTYH and its two interaction partners. And that resulted in these mice that had about 100-fold elevated germline mutation rate, including 99% C to A mutations. And when we compared the triplet contexts of those germline mutations in these mice to the odds ratios of mutation type enrichment in the D mice versus the B mice, we got a really nice match. So at the triplet context level, it looks like what we're dealing with is the signature of MUTYH, sort of confirming our intuition that this is the gene responsible. And moreover, MUTYH also has a known role in human cancer, where there are some families where people inherit pathogenic mutations in MUTYH. And it's sort of a recessive phenotype, but people who inherit either a homozygous pathogenic mutation or a compound heterozygote, they tend to be at elevated risk for colorectal cancers and develop this disease called familial adenomatous MUTYH-dependent polyposis. And what I'm showing you here is a correlation between our BXD singleton loading ratios and the composition of a cosmic mutational signature called SBS36, which has been recorded from somatic mutations in these patients who have pathogenic germline MUTYH mutations. And again, it's quite a good match. So there are actually 5 different missense mutations, missense differences between BLAC6 and DBA2J in MUTYH, and we don't at present know which one of these is most likely to be the culprit, or if perhaps there's some regulatory variant we're missing that might be the culprit. But I'm going to walk you through sort of what we've been able to figure out by looking at these variants in particular. So all 5 of these differences are predicted to have moderate impact by the SNP-F program. None of them is sort of a smoking gun that's high impact or homologous to any of the human pathogenic MUTYH variation. But what we did see that's exciting is that when we looked at genomes from some wild mice published by Bettina Haar, we did find all 5 of these variants to be segregating in wild mice, specifically in musculus domesticus, which is the species from which most lab mouse ancestry was originally derived. So this plot is just showing the columns are the 5 different variants in MUTYH, and they're colored sort of blue for the DBA2J allele and white for the C57 BLAC6 allele. And so you can see like this outgroup musculus like is fixed at these sites, but domesticus has variation at all of them. And what's interesting is that like musculus, it's this 2 million year diverged outgroup that is mostly reproductively isolated from the musculus subspecies. And so this would appear to be a good outgroup for calculating ancestral states. And what you see is that in 4 out of the 5 of these cases, it's actually the DBA2J alleles that appear ancestral and not the C57 BLAC6, which BLAC6 is the mouse reference strain. And so I think you often tend to just assume that the reference strain alleles are ancestral when perhaps you shouldn't. But I mean, more than that, it's kind of surprising that the BLAC6 variant may be derived, because then it's an anti-mutator rather than a mutator, meaning that the derived allele somehow improved the function of MUTYH rather than disrupting it. And so if we look at lab mouse mutation spectra from all the different strains in Beth Dumont's paper, we also find variation where, a few of the strains share 5 out of 5 of these alleles with DBA2J. Several strains share all of them with BLAC6. And then there's a group of strains in the middle that share 3 out of the 5 alleles with DBA2J. And then when we tested just we did a chi-squared test comparing the mutation spectra of each of these 3 groups of lab mice, we found a significant difference between sort of the 5 of 5 and 3 of 5 groups, and no significant difference between the 3 of 5 and 0 of 5 groups. And that provides, we think, some evidence that maybe one of the second 2 mutations in the ARG24 cis or the SEER69 ARG are that one of those might be the most likely causal variant. And then these heat maps just show you a comparison of the sort of log odds triplet enrichments in the D2 versus B6 BXGs versus the 5 of 5 and 3 of 5 lab strains that of course, the data from the lab strains is quite a bit noisier, but it does show the same triplet context as enrichments as the that we've been seeing all along, sort of suggesting what we are seeing in these lab strains is the action of the same mutator. And we also did a multiple sequence alignment of MUTYH in a bunch of other species, which sort of confirmed the idea that most of the alleles are ancestral in DBA2J and derived in C57 black 6, including the two that the lab strain analysis shows are most likely to be causal. And so yeah, what we think is going on is the discovery of an anti-mutator, which yeah, that basically just provides support for this theoretical idea in the literature that I'll go into a bit more later, that perhaps evolutionary pressure to optimize DNA repair enzymes is a bit limited. So I wanted to mention also just an extreme outlier strain, which provided a bit more interesting insight into what's going on in the BXD. So this is the same plot I showed you earlier of the distribution of C to A singleton fraction in the mice with the D haplotype versus the B one. And you'll see that there's this big outlier point in the Ds that's it ends up having this C to A fraction that's about 6.7 standard deviations above the mean. And when we looked further into this strain, it turns out to be only one of two strains out of all the BXGs that have a homozygous singleton in a DNA repair gene. And that gene turns out to be MUTYH. And then the singleton in this, it aligns to a site where human mutations are pathogenic. So basically what we think is going on here is that this BXD strain basically had a spontaneous mutation that's just like the ones that cause MUTYH dependent polyposis in humans. And then it has this extremely skewed germline mutation spectrum. So this is just to show you how BXD68, it has the highest estimated mutation rate of all the BXDs, about twofold elevated above the mean. And yeah, basically this is just evidence from Provian and SIFT programs that this mutation is deleterious to the murine MUTYH sequence. And when we actually did a calculation about the excess genetic load predicted to be caused by this mutator causing a twofold higher mutation rate, we think it actually is in the realm where it would be strongly selected against in mouse populations. Maybe you would have a selection coefficient around 10 to the minus 3, which is strong enough to be eliminated in an outbred mouse population. And we do have some anecdotal evidence from our collaborators that this strain is a bad breeder strain that appears to have some level of decreased fertility, which we don't know if how much that's attributed to this mutator versus other stuff that accumulates in the inbred mice, but it's interesting. So what are some future and ongoing directions in this project? I mean, we really want to do better possibly with like getting an experimental collaboration going to figure out like once and for all what the QTL maps to, and if it's one of these 5 missense variants on the DBA2J haplotype that segregate in the wild populations. And then we also we want to continue using natural variation around the QTL to determine how much activity this is having in the wild mouse populations. What we have so far basically indicates that this isn't like a dominant cause of mutation spectrum variation in wild mice, but it looks like it might be just one of many mutators around that shape variation, and just we happened to discover it because of which strains were picked to make the lab mice back when that was done. And then yeah, we think basically that this mutator should be well under the nearly neutral threshold. And so I mean, it remains to be seen whether there are any somatic disease phenotypes linked to it. And then after we control for variation at this locus, we're interested to see if there are any other modifiers of mutation spectra specifically in the BXD where we could find them. But I mean, potentially this strategy could also be used to look for mutators in other recombinant inbred lines that haven't been inbred for as long if more offspring are sequenced to yield a similar number of de novo mutations. And then we also want to know, of course, what's the, now that we sort of have some evidence for MUTYH variants having germline mutagenic activity in mice, like the natural implication is if MUTYH variation is also mutagenic in the human germline, and like this links to this broader question of like how much is the genetic architecture of say cancer risk variation in humans really bound up with this variation that we can see in mutation spectra in the germline. So with the time I have left, I just want to tell you about a second story we're really excited about of looking at for natural mutators in the yeast Saccharomyces cerevisiae. And the way that we were doing this was quite different. Instead of leveraging, say, lab crosses of S. cerevisiae, we went and looked at this really nice panel called the 1011 yeast strains, which was assembled a few years ago from a combination of domesticated and natural environments. And basically, when my postdoc, Penyao Zhang, did mutation spectrum PCAs of all these yeast, we saw this kind of structure that we're now sort of expecting to see because we've seen it everywhere we've looked for it, of differences among the different populations that suggest that mutational processes have different biases in the different yeast lineages. And again, we had this question of how much of this variation, which is inherited variation, is driven by genetics versus past and present environments. And again, we're well suited to answer this question using yeast because yeast are easy to grow in the lab. And when we measure mutations in that controlled environment, we can isolate just one of these variables. And so working with Maitreya Denham and her student Anya Oladart, we developed this technique, which is again, sort of like an updated version of a very classical genetics technique using what's called a CAN1 reporter gene assay to detect, to like efficiently detect mutations accumulating in these different yeast strains in a lab environment. So the way that works, which I'm sure any yeast people in the audience know, but others might not, is that when yeast with a functional canavanine transporter is grown in the presence of a drug called canavanine, the transporter imports the canavanine, which is sort of a toxic analog of arginine, and then the cell dies. But then if you happen to have a spontaneous mutation in CAN1 that disrupts its transporter activity, then you get a resistant strain, which you can grow in the presence of canavanine, and it doesn't die. And so then this is useful as a reporter gene because you can grow a bunch of yeast, let them accumulate spontaneous mutations all over their genome, then poison them with canavanine, and then isolate some yeast that all have mutations in just one gene that you can sequence for less money than the whole genome. So what we did was basically the first step is just to do a classical Luria-Delbruck style fluctuation assay where a bunch of yeast are grown up, and some of them will spontaneously get loss of function CAN1 mutations, and then you impose a selection with canavanine and grow up the yeast, and then all these colonies will be mutants. And so the number of colonies is classically used to estimate the mutation rate. But then we went a step further of growing up the mutants and then sequencing the CAN1 gene, and then getting a mutation spectrum of the variants from there. And so this is what we got from a sampling of haploid strains from the 1,011 genomes. Basically, this assay only works well on haploid strains, so that limited our options a little bit, but still gave us a bunch of diverse strains to work with. And so these are histograms of like how many A to C, A to G, A to T, and so on mutations there are in each of these strains. And you can see that the two strains at the bottom called AEQ and AAR are highly enriched for C to A, which is an odd coincidence with the mouse project. That was very fun to see. But yeah, like these strains are related to each other. They're both classified as mosaic beer yeast. And they actually have about a 10-fold higher mutation rate than the other yeast, as well as the skewed spectrum. And this is just showing their mutation rates compared to the others in our assay. And then if we look at, if we do a PCA just of the mutation spectra of rare variants across these yeast, then that makes the mosaic beer yeast pop out. It's a bit hard to see on here, but the mosaic beer yeast are all of these ones that are represented in green, and they include AAR and AEQ and several related strains. And so we think this is evidence that the mutator that we can see being active in the lab has also been active in these strains in their natural environments in recent history, and is causing some of this variation that we see in the mutation spectrum PCA. And really, we wanted to use this sort of as a proof of principle that some of this variation that we see in these plots really is caused by genetic mutators. And I think even without knowing what gene causes this in the mosaic beer yeast, this shows that there does appear to be an active genetic mutator causing this pattern. And there's two phylogenetic sister lineages to AAR and AAQ that appear to have similar mutation spectra, even though we weren't able to measure their mutation spectra in the lab. So this is just a little close-up of the phylogeny with sort of the green strains that we want to look at. And basically, if we sort of plot the C to A fraction as a function of allele frequency in these strains and these red outgroups, we see this enrichment in the mosaic beer strains at low frequencies, suggesting a recently emerged mutator. And then this has been replicated actually in a couple of different data sets, both the 1,011 yeast genomes and then a data set specifically of beer yeast. And then just looking more broadly across the yeast phylogeny, we see like this mosaic beer yeast clade sort of being one of the clades that sort of have clustered elevated C to A mutation rate. And a really exciting thing is that this clade actually contains a missense mutation in the gene OGG1 relative to the standard lab yeast, and OGG1 is actually an interacting partner of MUTYH in the same 8-oxo-G repair pathway. So we're still trying to verify whether this mutation is in fact responsible, but basically we went through and annotated, what are the variants in DNA repair genes common to AAR and AEQ, and then how many of them meet the criteria of being present in both of these, having relatively low allele frequency, and not being present in any of the strains that we measured to have normal C to A mutation fractions. And that, yeah, one of the only genes meeting this criteria is this OGG1, which is also the one expected to have this C to A signature. So what I wanted to leave with as sort of a takeaway from both of these stories is that we think that like these two different natural mutators together support this drift barrier hypothesis that's mostly been like a compelling feature of evolutionary theory, but like until recently hasn't had much data to support it. And basically what I'm showing you here is a supply and demand curve of the idea that sort of on the x-axis is you spending more resources to avoid mutations, to repair your DNA. And that's sort of represented by the blue curve, your cost of fidelity going up. And at some point, like the amount you're spending to avoid mutations is going to exceed the cost of just incurring those mutations. And the intersection of those is this theoretically optimal mutation rate, which Michael Lynch calls the biophysics barrier, sort of what's the most accurate optimal mutation rate that you can get. But what Lynch actually hypothesizes is that most populations will exist at mutation rates above this optimal mutation rate. And how much above depends on the effective population size. Because basically, if you have a mutation that increases, or if you have, say, a mutator allele that increases your mutation rate, but the excess mutations that you get from that only give you sort of an excess mutational load of sort of fitness cost 1 over the population size, then that's going to be nearly neutral, and you're not going to be able to select away. You're not going to be able to select that out of your population, and so this will naturally just drive mutation rates to be up to this drift barrier. And a consequence of that is you kind of expect to see these mutators commonly segregating in populations, perturbing mutational spectra, and even in the case of if our hypothesis about mouse MUTYH is correct, leading proteins like MUTYH to just sort of have functions that are suboptimal enough that just like 1 point mutation or maybe 2 can lead to a noticeable improvement in their efficacy. So I'd just like to thank my lab and funding sources and collaborators, and then I'm happy to take any questions.
I'd actually like to start with a question I had about so with the MUTYH, and I guess both pathways you talked about because they sort of do the same thing. Do you know if there are any environmental factors that would increase the rate of that type of damage that MUTYH is repairing?
Yeah, that's a great question. I think in the case of yeast, like that absolutely could be the case, because yeast can either do oxidative, they can either do oxygen respiration or they can do fermentation. And that's why like yeast can be used to like make alcohol in beer and wine. And I mean, oxidative stress is maybe greater in environments with oxygen. So I think in the case of yeast, like I've wondered whether it's significant that like we observed this in a beer yeast, or maybe like the pressure to avoid oxidative stress is a bit relaxed. I'm less sure in mice, you know, at least in the wild. I mean, maybe high altitude mice could have less oxidative stress, but I'm not as sure in that case. I mean, maybe there would be sort of different metabolic phenotypes that would increase that would cause oxidative stress to vary within tissues, though.
Thanks. Does anyone else have any questions?
Yeah, this is David Rand. I had a very simple question. Is there any sign of unusual levels of variation around your mutator or anti-mutator locus in most populations? Maybe you would describe that from the comparison of those 5 strains, but
no, we didn't really, yeah, but that's a great question. I mean, we sort of ran a DNDS analysis to see if there was any evidence for positive and negative selection, and we didn't really see that. And when we did our multiple sequence alignment, I mean, it looked like sort of a gene with like a medium amount of conservation, especially in like the sites we look at. But I think like overall, there aren't really signs of it evolving in a particularly unusual way that we can detect yet. And that would fit with sort of the hypothesis that this mutator or anti-mutator is basically evolving neutrally. But I mean, what would be really nice is if we could see a signal of whether mutations were accumulating slightly faster or not on like in lineages that have one mutation or the other, but the effect is so slight that in an outbred population, like a lot of the new mutations that would be created by the mutator or avoided by the anti-mutator, if they occur far away in the genome, that really limits our power to see they're not linked to the variant that's sort of causing them or avoiding them, and that's sort of what makes the inbred system so much more powerful for discovering loci like this.
Right. No, the distal mutator effects are probably more evident. So, thanks. Nice talk.
Oh, thanks. Oh yeah, I think Kelley has a question.
Hi. Yeah. I was wondering, since we know you presented towards the end that there's this effective population size on kind of the tolerance for mutators, I wonder if there was any evidence of the effective population sizes, either of the mouse lab strains or the yeast strains, that kind of maybe correspond, or if you've looked at that at all as something influencing.
Yeah, we've definitely been looking to see if we I think what this hypothesis would predict is that the more sort of the smaller NE is and the more a population is dominated by drift, we might see sort of accelerated emergence of mutators and more neutral drift of them, versus if it's a population with a really large effective size, we might see a more constant mutational spectrum. And I guess that's a pattern that you need a lot of data points to be very confident of. It's something that we've been trying to accumulate data on as we do these analyses in more organisms. I think with the yeast, they're probably the largest NE of the populations we've looked at, and you do see that the yeast look a lot less dispersed in their PCA plots than, say, the humans or the mice do, especially when you control for how far genetically divergent the yeast are. So we think that just the difference between the yeast and everyone else tentatively supports that prediction, but we'll have to see as we add more organisms if that seems to hold.
Any other questions? All right, well, thank you so much, Dr. Harris, for joining us and sharing your research with us this afternoon. And thanks to everyone who attended. I hope everyone has a nice afternoon.
Yeah, thanks very much for the invitation and all the good questions.